Hello there. This video is about gametogenesis, which is the process of making sperm and eggs. We've spent some time looking at the anatomy, the internal anatomy, the external anatomy. We've looked at how it forms from the embryo into the adult. And all of that anatomy is simply to support these two cells meeting. So the human sperm, which is the smallest cell in the body, and the human egg, which is the largest, and this is more to scale than the previous image, have to meet in order for our next generation to exist. Now, remember that our cells have two sets of cookbooks. By cookbooks, I actually mean chromosomes, which contain DNA, which contain the genes to make which are the recipes to make proteins. But let's just work with the cookbook analogy for a moment. So we have two sets. Each one has 23 volumes, and one of those came from our mother, and one came from our father. So that means that we have 23 pairs of homologous cookbooks. Homologous means that it's the recipe for the same dish, or in this case, the gene for the same protein. However, there are variations in the recipes, which are called alleles. So to stick with the cookbook analogy, if there's a recipe in this cookbook for oatmeal cookies, then that same recipe is also in this one because they are homologous. However, this oatmeal cookie recipe might be the allele that calls for chocolate chips, whereas this one may be the allele that calls for raisins. So we have 23 pairs. This is a pair, a homologous pair of cookbooks. All right. Now, sperm and eggs only have one set of cookbooks apiece. And that's so that when they combine, our sperm with 23 chromosomes or cookbooks can combine with the egg that also has 23 cookbooks or chromosomes, and that's going to give us a zygote, which is what we call the cell once the DNA has mixed together again. And that zygote now will have 23 from the father, 23 from the mother, a total of 46, or 23 pairs of homologous cookbooks or chromosomes. And so this is once again a homologous pair of cookbooks. Now, sexual cell division, which is called meiosis, is the process by which we end up with daughter cells that only have one set of cookbooks. So this is how we have to make sperm and eggs, because if we didn't get down to one set of cookbooks or chromosomes, and we had a sperm with, say, 46 and an egg with 46, then when they actually combined, it would produce a zygote with 92 chromosomes, which is way too many to be able to function. Now, there is a term for cells that only have one set of cookbooks, and that is haploid. So the whole point of meiosis, or sexual cell division, is to produce haploid cells, or cells with half the number of chromosomes than the adult or parent cell. Okay, now, of course, we're talking about chromosomes and not cookbooks and all of this. So let's look at this again. Here we have a cell that has one pair of homologous chromosomes. I'm not going to draw all 23 pairs because that would be impossible to follow. So we're just going to look at what happens to one and understand that it's happening to all of them. So this parent cell has one paternal chromosome, or one from the father, and one maternal chromosome, one from the mother. They have the same genes, but they may have different alleles. And sexual cell division is going to allow these to divide to produce daughter cells that each have just one chromosome and not a pair. Okay, so the cells in the ovaries and or testes start by making a copy of every chromosome. This is so that we can get more bang for the buck. We can produce more cells with each cell division. So this chromosome makes a copy of itself, which is here. So this is the original copy, and this is the new copy. Not really, but close enough. And they attach to each other by a part of the chromosome known as the centromere. But just think of them as being two identical copies stapled together. And each of these copies is called a sister chromatid. So, what happens when the cell is getting ready to divide is that the chromosomes line up next to each other in the cell. So this pair is lining up here, but the remaining 22 pairs would be doing the same thing, all of them in a long line. Again, it's too much to draw in one diagram. Now, how they line up is entirely random. So it could be blue on the left or blue on the right, and that's going to be the same for all the pairs of chromosomes, which means that this is one area where we generate diversity in the sperm or the eggs that are produced. Now, once they're lined up like this, sitting next to each other, they're going to swap some genes. Because they have the same recipes or the same genes, it's actually fairly easy for the DNA just to switch chromosomes. And so now, one of these sister chromatids, though it's mostly paternal, will have some maternal DNA. And one of these sister chromatids, even though it's mostly maternal, will have some paternal DNA. This generates even more diversity. Now, this cell then divides. You can see it beginning to split here. And each one of those daughter cells gets one of each chromosome pair. Right? So we have the mostly paternal one here, the mostly maternal one there. Now, each one of those is going to then divide, but this time, instead of separating chromosome pairs, we're actually going to separate the sister chromatids, the two what used to be identical copies, or at least they were identical before they did the DNA swapping. And once that division is finished, we have 4 completely unique daughter cells. Okay, so here are our 4 unique daughter cells. And the way that they become sperm or eggs is different. Now, the DNA, the cell division that we just looked at, is the same for sperm and eggs, but they develop very differently, not just how they develop, but when they develop. And furthermore, they are both controlled by the same hormones, so we're going to look at each of those.